everyone. Good afternoon or good morning, depending on what time zone you're in. I'm going to introduce our speaker today, who is Dr. Renee Deehan. Renee Deehan and I are both calling in today from sunny Massachusetts. She has spent her career in industry, extracting insights from omics, clinical, and patient-generated health data. She served as Chief Scientific Officer at Solventa and was the VP of Biocomputing at Patients Like Me. Most recently, she was VP of Computational Biology and Translational Informatics at Precision for Medicine. Dr. Deehan received a BSE in bioengineering from the University of Pennsylvania and a PhD in molecular and cell biology from the University of California, Berkeley. Thank you so much, Dr. Deehan. I'm going to turn it over to you now.
All right, awesome. So I am going to fix my screen first because I bet you can't see it all. Let's try this. Can you see it now? No. Well, yeah. Okay. All right, so actually, let's start with the agenda first. We'll do an introduction and then kind of delve into the science, both from a methodological perspective as well as an application perspective. We've taken a lot of different computational approaches, but what I'm going to focus on today is the application of prior knowledge, as Anne said, to interrogate multi-omic data using this approach called reverse causal reasoning. And then after that, and a lot of the work that I did at Solventa was using this approach. And after that, I'll talk a little bit about Digital Me, which was the work that we did at Patients Like Me, where we were trying to kind of digitize patient biology and the patient experience alongside with molecular data. And then we'll talk a little bit about future directions. So, I'm going to start with this meme, which I'm sure many of you have seen, and it starts out with the relationship between data, information, knowledge, insight, wisdom. So you start with data. If you are able to add some context to that, you can generate information. If you're able to make connections between bits of information, that's kind of knowledge. If you're able to, at least in the version of this model I'll be talking about today, if you're able to distinguish signal from noise, using that knowledge, we'll call that an insight. And then, really making a decision using that insight might be called wisdom. The joke here is that the next stage after that is conspiracy theory. And if you want to, you can look at a pattern of data and you can find anything. And so when I first saw this meme, I thought it was just delightful because, of course, it's capturing information and knowledge in that whole relationship, but then also conspiracy theory, because I'm convinced that this is what the more quantitative data scientist types think about biologists. And I'm sure that we've often heard people say, and I have been on both sides of this equation, that biologists can literally make a story out of anything. And you can hand them a list. It could be a random list of genes that might describe, colon cancer, and they're still going to be able to, make a story out of that. And I always laugh because I think there's a really good reason for that. And that is that, biology is insanely complex. And so, here's an example of some of the richness and the complexity that biologists are trying to work with. So on the left-hand side, it's the classic kind of updated Hanahan and Weinberg hallmarks of cancer. And in the upper left-hand corner, you can see this green module right here, which is sort of this general process of increased cell proliferation. And there are other modules, I'd say biological processes here, kind of higher order biological processes like dysregulation of angiogenesis or inhibition of apoptosis. But if we just go back and we look at this kind of cell proliferation cartoon over here, what underlies these, 4 green blobs is something with the complexity of this, which this is a manually curated network describing cell proliferation, and there's 848 nodes and about 1,600 different edges. And it's called a hairball, which is just a terrible description. But this hairball could also be called a big ball of conspiracy theories. And what the biologist is doing when they get that list, of course, is they are taking that, let's say, output from a statistical model or another type of model, and that's information or knowledge that they have, but they are contextualizing those results in their own head, using their own knowledge network. And of course, the biologist can't, or most people that I know are not able to hold this much information about cell proliferation in their heads and then sort of generate insights from it. But biologists do, especially signaling biologists, they really do hold a lot of information in their heads. And so when you hand them a list of genes or mechanisms or something like that, there's oftentimes many different answers in terms of what the interpretation of that is. And so that's why I think they're seen as these conspiracy theorists, when I think they're really just trying to be quite clever about their application of those results to their knowledge network. And there's just a lot of complexity there. So where we are in data life today is that really the pace of our ability to generate data that can inform more and more of those conspiracy airballs is the pace of that is pretty unrelenting. So when we say multi-omics these days, usually it refers to DNA and RNA-seq first and foremost, maybe some proteomics, maybe some functional data, maybe some imaging data, but also copy number data, metabolomics, lipidomics, et cetera. Methylomics is a great one. And a great example, I'd say probably one of the consummate examples of large-scale data that we have access to now is the Cancer Genome Atlas, which I'm sure all of you are familiar with, where there's sort of multi-omic profiling of 11,000 patient tumors. Some of them even have adjacent tissue controls. It's so like 2.5 petabytes of data. I mean, it's just extraordinary. And what's pretty awesome and wild about it is that you can just have this for free. You can just go and download, download all of this and analyze it. And this is pretty amazing because when I started out 15 years ago, it was really difficult to get your hands on data. And I'd say actually the quality of the data was very low. And we used to joke that when you downloaded a data set from Geo, the annotations were more likely to be like a list of Burger King menu items rather than like tumor stage or something useful like that. But of course, the challenge that we're dealing with is that we are actually better at generating data than we are at extracting value or extracting insights or wisdom from that data. And this is a figure from a review from Rossi and Griffintini titled Big Data, Challenge and Opportunity for Translational and Industrial Research. And really, it's just making the point that our ability to interpret this data has not caught up with this orange line here, which represents the amount of data that we are dealing with. Actually, I think they call it data per decision. And the blue line right in the middle here is the limit of human cognitive capacity. I actually think that's a pretty generous intercept. And the point that they make in the review is that we're really going to have to augment our own kind of cognitive capacity with what they call actionable knowledge. And so, the question is how are we going to do that? And the answer is artificial intelligence, of course, and that's it. We're done. I will take any questions. Just kidding, of course. But I make that joke because I feel like artificial intelligence right now is like the cause of and solution to all of life's problems. But the real meat here is digging into, well, how are we going to actually apply artificial intelligence to these problems in order to help accelerate our ability to generate those insights and really kind of close the gap between data generation and insight generation. Now, oftentimes when we say artificial intelligence, probably most people think, most people not being this crowd, but most people associate this with machine learning. But the main focus of my research, our research, has been in the other, another type of artificial intelligence called knowledge representation and reasoning, the other AI. And this is a very simple example of what that might look like. And I'm using Judea Pearl's raining example here, where we may have in the middle of this is there's an agent, and this is the reasoning engine. And in this example, let's say the reasoning engine is me, and I need two ingredients in order to be able to reason. I need data, and I need a knowledge base. And in this case, the data is the ground is wet. The knowledge base, which is what is known, is that raining causes the ground to be wet. And I can now reason, do some forward causal reasoning, and deduce that, in fact, it is raining. And from that, I can make a wisdom decision where I will wear my rain boots outside. I know, absolute genius here. But if I walked into your office and I was wearing rain boots, you may do some reverse causal reasoning, which is, Renee is wearing rain boots, it must be raining. So very simple examples, but very analogous to what we are trying to do on the molecular kind of interrogation scale. So up here, this multi-omic data would be, what the reasoning agent actually senses. The knowledge base is really a collection of, cause and effect relationships or correlative relationships between, information, things that we know. And the reasoning engine can generate some insights from it. And then, in our definition of this for today, really the wisdom bringer would be the biologist, the clinician, but somebody who is using what the reasoning engine kind of produced in order to make an actionable decision. And the types of actionable decisions that we've been working on, pertain to the applications of drug discovery and development. Of course, these are real kind of like industry problems. And these are just the classic personalized medicine problems. But a lot of the work has been done to build mechanistic models of disease, understand the mechanism of action of drugs and maybe off-target effects, or compare, we would call it competitive intelligence, where we would be looking at one company's drug and comparing it against the standard of care, identification of targets, being able to discover biomarkers to stratify patients, et cetera. The same types of problems that you all are working on. So I will now move into the approach of causal reasoning. Maybe pause, see if there's any questions. Probably not. Cool. All right, so I'm going to tell you about two different things, those two ingredients that we need in order to reason, actually. Just one of them. We'll talk about knowledge representation in building a knowledge base, and then kind of the algorithm itself for reverse causal reasoning, and then some applications of it. So, and what we're going to talk about first is a purely qualitative method of doing reverse causal reasoning. So this is an example or a description of the knowledge base that we had at Solventa, and I consider the knowledge base the intelligence collective. So what we were doing is, extracting cause and effect relationships from the literature. And if you think about it, I mean, it's just, I love this. Like, I love just thinking about how much wasted knowledge is sitting in PubMed, in PDFs, just waiting to be used and extracted. And I know, obviously, like Larry et al., people are doing some really beautiful work, automating this element. But this particular knowledge base that we had at Solventa was actually manually curated, or if we used an automated extraction process, it was always like manually reviewed. It was fairly large for a manually curated knowledge base. I think at the end we had over 600 causal statements, tens of thousands of manually curated publications, and the content of that was largely focused on the work that we did with our clients in pharma and biotech. And I think it is very fair to assume it's oncology and autoimmune disease, and then like some other stuff in there. So probably 60 or 70% oncology and the rest autoimmune and other. And the way in which we would capture this information is using a language that was developed by a scientist by the name of Dexter Pratt, who is running the index project over with Trey Eidecker at UCSD. And really what he attempted to do and what he really achieved was building a language that was able to represent, kind of what I would call high resolution biology, and kind of like functional relationships, cause and effect relationships between molecular entities. An example of that is shown here, where you may read a paper and there would be a statement in that paper like RNA expression of RBL2 is directly mediated by the activation of the FOXO3 transcription factor. And right here is this written statement written in the language of Bell. So it's computable, and it's also human readable, which is really important as well. So what this says here is the T-script, the transcriptional activity of the protein FOXO3, the namespace is HGNC, activates the RNA expression of RBL2. And this is a Bell triple. These triples are then assembled into computable shareable models for use in different types of applications. These triples can be annotated with body part, tissue, cell type, experiment type. There's a free text form in there for you to be able to write in whatever you want, who curated it, when they curated it. Of course, all this type of information is collected. And actually, one of my personal favorites about the Bell language was two things. First of all, just the real heavy emphasis on cause and effect relationships in biology, because that's the way we biologists think. We think in pathway diagrams a lot, but also that it did have that resolution that you could distinguish an RNA from a protein, that you could distinguish or label a kinase activity or a particular modification like an acetylation or a phosphorylation event like that. And then these very tight, high resolution molecular entities like this can also roll up into higher level processes like cell migration or cell proliferation. So before I dive into the kind of algorithmic part, reverse causal reasoning is really just a very fancy way of saying forensics or forensic reasoning. And the easiest, simplest way that I think about it is what you're seeing right here, where we have the dominoes, they've fallen over, you walk into the room, and it is your job as the agent to figure out who knocked over all of those dominoes. And you may have many piles and many who's that knocked over the dominoes, but really you are looking at the scene of the crime, which might be an RNA-seq experiment, and you're saying, who caused this? And you're using the knowledge base to do that. So, and I'm going to go through this fairly quickly, and we could talk later about any details, but an example of how this works is the following. This is the LNCAP cell line. It's been treated with this synthetic androgen. Microarray experiment was done comparing treated versus control. There are significant changes in gene expression that were identified. And those are what are highlighted here in the bright red and the green. In this particular case, it was an AFI array. There were about 1,200 measured transcripts. And of that 1,200, 691 significant gene expression changes were identified. So it's 5%, it's really, yeah, about 5% actually. And the next step is to interrogate these, statistically significant changes in gene expression against a knowledge base. So that's what we do. We take the gene expression changes and then map them onto the knowledge base, just like our biologist did earlier on. And then those map changes can lead to different types of testable hypotheses, and this really is reverse causal reasoning again. So we're looking at the pattern of gene expression changes, and really we're saying, all right, this is interesting. Can we scan all of the knowledge that we have in our knowledge base of all of the different other gene expression patterns that we know that can be controlled by upstream regulators, and see what our experiment matches, what it looks, most closely like? And I think a more kind of intuitive way to think about this is to think about these mechanisms. And in this case right here, we have the transcriptional activity of the androgen receptor. And what you're looking at is this directed acyclic graph that we have in the knowledge base that has been curated, describing the cause and effect relationship between AR transcriptional activity and downstream changes in gene expression. So what we have here is this particular directed network, and that becomes important later, like in 2 seconds actually, is this network has 268 downstream entities, right? And in this case, 268 RNAs. So there were a handful of papers actually that were curated where somebody perturbed the androgen receptor and then looked downstream after doing like probably a microarray experiment and identified 268 genes from either one or more experiments that were modulated when the androgen receptor was modulated. So out of the 268 genes that we know that androgen receptor can control, 44 of those genes were actually modulated in the experiment. So that's what you see with the bright greens and reds that are actually mapped onto this knowledge network for the androgen receptor. And out of those 44, 36 of them were changed in a direction that is consistent with increased androgen receptor signaling rather than decreased androgen receptor signaling. And there's kind of another metric in here, which is that, believe it or not, this actually passed an enrichment statistic. So out of the 268 RNAs that we know that the androgen receptor can control because we've curated that information, 44 of them were modulated in this experiment, and that is actually significantly more than would be expected by chance. So just like you would do in a GSEA type of analysis, there is an enrichment of state changes in this particular experiment with that androgen receptor drug, compared to what was possible. So imagine doing this at scale. So you're looking at the transcriptional activity of AR network. And in fact, there are thousands of these types of directed networks that can be interrogated with a data set that exists in the knowledge base. And so when we do it at scale, it might look something like this, where not only do we have AR, but also sometimes we'll see it called a hypothesis or a mechanism. EGFR, IL-6, PTEN, et cetera. There might be a couple of mechanisms in here that are predicted to be statistically significantly modulated, but we may deprioritize them for one reason or another. But now we can start to build a higher order causal network model of these inferences, these hypotheses, these mechanisms here by describing the relationships that connect them. And this is really the fundamental concept of it all. And while I will be talking mostly about transcriptional data, there are ways in which you can start to integrate other types of information into these. Proteomics is a really obvious one that we have explored doing reverse causal reasoning with in a few cases. The integration of other data modalities, honestly, right now is more of a post hoc. Either we would look at like a correlative approach to connect them into the network, or we would use our prior knowledge network and map, let's say, metabolomic changes alongside with all of these inferences that we might get if we were trying to build a model of drug response or drug effect. So just to kind of summarize, there's two different metrics, and if TNF is the example here instead of the transcriptional activity of AR, but richness is really evaluating, is there the presence of a molecular signal in my transcriptomic data for TNF pathway signaling? Concordance is really saying what is the direction of that molecular signal, which is, do I predict that in my experiment is TNF increased in abundance or activity or decreased in abundance and activity? And then the third thing, which I'll talk about in a little bit later, is strength, which is kind of it's a riff on this qualitative approach that allows us to get a relative sense of the amplitude of TNF signaling, in one condition compared to another. I'm not going to talk about translatability or larger network scoring in this talk, but I think it's important to raise some of both the strengths and the weaknesses of this approach. And we'll start with the strengths. So it is able to infer the activity state because of the way the knowledge has been curated. It infers a direction of that activity state. It can be semi-qualitative. That's what we call kind of this amplitude strength scoring. It can uncover relationships that were perhaps studied in one context, but not another. So it can discover new biology that already exists. It can be forgiving with respect to data variability. And what that means is, you and I might have actually equivalent levels of TNF signaling, but the gene expression pattern that describes your TNF signaling is different from my TNF signaling, and that may or may not be important. It is actually great for smaller data sets because you're able to constrain things with a knowledge base, and it's also white box or explainable. Definitely some limitations, so this is why I consider it an approach that is complementary to other computational approaches. So some of these limitations are that it actually requires a knowledge base, and there are not knowledge bases that are, just readily available that have been structured in a way that are, competent for reverse causal reasoning just everywhere. Knowledge bases themselves are, of course, they're absolutely biased in multiple ways. So they can't capture what hasn't been studied by humans. They, in this particular type of knowledge base, where we're extracting information from the literature. And it's biased by what topics have gotten research funding, what people have decided to publish. People don't publish negative data, for example. It's biased by also what we, for example, at Solventa, decided to curate from that pool of knowledge that exists in PubMed, et cetera. So, and it actually cannot discover entirely novel relationships because it is constrained by that known knowledge base. And, a complaint of mine would be that it actually doesn't handle temporal data very, very elegantly. If you have a longitudinal experiment, you can definitely build a model at each time point. But, kind of seamlessly connecting those models across time is not something that just does well. It's not a dynamic modeling approach. I'm going to skip this one because we probably all know about white and black box, but my favorite way to explain black box is that it's the sorting hat, because the sorting hat makes decisions, but we do not know how the sorting hat comes to their decisions. Okay, so just some applications of this type of reasoning. First of all, just review a typical causal modeling pipeline architecture. So we may have, different types of data here, clinical, DNA, RNA-seq, epigenetics, proteomics are probably the most common that I've dealt with. There's always a process of data integration, quality control, transformation, and then after that, that is transformed into what I would call a data set that is analysis ready. So you may have extracted only the data points that you need, and they have been appropriately transformed to be able to go and do whatever, mathematical modeling approach that you want to take. And then also there's the knowledge graph that we have, and it's this combination of analysis ready data and knowledge that is fed into the reasoning engine. And what we're talking about here are knowledge-driven algorithms, but we can also use other algorithms here that are entirely correlation-based or data-driven. The output of this is what I would say is a raw computational model. I would say that this is an insights. This might be like a list of potential explanatory hypotheses, like the transcriptional activity of AR for the data. There's a step after that where we actually have to do a lot of, and this is actually the longest step, is QCing those insights, those inferences from the robot. And then after that, there would be the implemented action of wisdom. And then of course there's this line that you can't entirely see, but that goes from implemented action to, back to data, because everybody really wants to believe that they're working in a learning loop where you use data to make a decision that then drives the generation of new data that you can kind of put back into the system. And that is the goal. So one example of this qualitative approach, and this is work that was done in the last couple of years on COVID, and this was an early microarray data set, early RNA-seq data set that was made available, I think 2 Mays ago. And this group, they published data that looked at transcriptomics and some other data modalities, but we only looked at transcriptomics from human lung biopsies, people with COVID, also people without COVID. So we had healthy controls and could do a differential gene expression analysis. And they were also looking at different cell line models of COVID and also a ferret model. We didn't look at the ferret one yet, but that would be pretty cool. But they had 2 different cell line models that they were using. First was NHBEs, and they would infect SARS-CoV-2 and compare to non-infected. And then they also had an A549 model. And the A549 model actually doesn't express ACE2. And so, which is the receptor for SARS-CoV-2. And so what they did here was they overexpressed ACE2, and then they added SARS-CoV-2 so they could actually infect the A549 cells. And the last kind of contrast that they had was this A549 model, where they also treated those cells with ruxolitinib. So they were really kind of looking to first explore cell line models of COVID pathogenesis and also, the impact of a potential, therapeutic. And this is the results of our reverse causal reasoning, our qualitative reasoning analysis of that. So for each of these 4 different conditions, we would generate, do a differential gene expression analysis, identify significantly modulated state changes, map them onto the knowledge base, perform reverse causal reasoning, and then, the output would be kind of listed out in a format like this, where we could look and say, okay, well, the top one here is a hypothesis saying there's a decrease in CD40 ligand predicted. And then underneath that, an increase in interferon alpha family signaling, and then in decrease in the transcriptional activation of HIF1A, and so on and so on. And you can see that there's different types of entities in here, not just molecular ones. There's things like tunicamycin, which is an inducer of ER stress. And what this means is not that these COVID patients have ingested some tunicamycin. It just means that it may be a proxy for decreased ER stress response. So the color pattern that you're going to see here is yellow is a predicted increase, blue is a predicted decrease. The intensity of the yellow or blue actually corresponds to it corresponds to concordance, which is our confidence in the predicted direction of that mechanism. So what is actually quite nice is that this is just a very kind of easy and intuitive way to start comparing and contrasting different mechanisms. And remember, we're contrasting these, we're looking at these at the mechanistic level, not at the gene expression level, which actually I forgot to mention this before, but I think one of the real key points of reverse causal reasoning is that it is not forward causal reasoning. And what I mean by that is that a lot of computational approaches will, knowledge-driven computational approaches will take, a knowledge graph like a KEGG pathway, and you'll get a list of genes in your experiment, say infected versus not, and you map those gene expression changes onto this pathway and do an enrichment analysis. And you can then kind of rank these pathways, but the assumption there is that the increased abundance of mRNA is 1-to-1 correlated with the increased abundance of proteins abundance, which is also 1-to-1 correlated with an increase in protein activity. And we just know that that is inherently false. And I don't say that because I do not like these other methods. I do when I use them. I actually like to have both, but it is not a guarantee that just because you have an increase in mRNA expression that that's going to functionally translate to an increase in the activity of that gene. So what we're looking at here is this middle column here is the COVID model from the human lung. And on the left-hand side, this was the NHBE cell line model. And on the right side, this was the A549 cell line model where they overexpressed ACE2. And you can start to get a very kind of fine-grained mechanistic view of what's going on in one cell line over another. And this type of mechanistic view helps you make decisions about which cell line you're going to move forward with. And this is something that's actually a very kind of tangible problem for folks in industry. They have a finite budget and a finite amount of time. And so making the appropriate decisions about which cell line model to use rapidly can help them save that money and time. And I think actually even much more importantly, it can, potentially, hopefully mitigate failures in the clinic when those failures are due to the lack of translatability between an animal model and the actual human condition. So, the last kind of build on this I'll show you is on the right-hand side here. This was the model that was generated with the ruxolitinib treatment. And, really what you can see here is, here's the human model right here. We have our 2 cell line models on either side. And, which largely actually in this case do recapitulate some of the biology that's seen in the human, or a good portion of the biology, the surprising portion of the biology seen in the human, of course, you're going to be missing things like angiogenesis, et cetera. Here's an example where it's a little bit different in the cell lines. So the cell lines have an increased response to hypoxia, whereas the human did not. But what you're looking at here is the ruxolitinib treatment, and you can see that a lot of this disease biology that's present in the cell lines and in the human is then resolved in the cell line when you treat with ruxolitinib. This is kind of like the interpretation of some, holistically, the list of hypotheses and mechanisms that we were investigating. And we saw a lot of type 1 interferon, TLR7, IRF5 signaling, and these were findings that were consistent with some of the clinical findings in COVID-19 patients. The interesting one was the decreased B cell signaling. And I know that this data set has actually been updated since, so it'd be really kind of cool to take a look at it and also compare the ferret data that they had, because apparently ferrets are a better COVID-19 model than mice. So I am going to actually skip this because it's I think you guys get it. But like I mentioned before, we do have like a riff on this method to be able to infer mechanism amplitude or strength signaling. And the way that this is done is essentially what we call it semi-quantitative reasoning. So in this particular case, there is no conflict, I'd say, between the predicted direction of this mechanism and the downstream genes, meaning that the knowledge base says that an increase in this mechanism should yield an increase in gene 1, and in fact, that's what we see in the data. So in this case, what we can do is we can essentially just summarize all of the fold changes from these genes, and do that on a sample by sample basis. So in this case, we actually wouldn't necessarily need to, and we're not doing a statistical analysis identifying significantly differentiated genes in a case versus control scenario. You can actually just take a bunch of tumor samples and compute fold changes off of a centroid or a meteoroid or something like that, whatever you want, generate these fold changes. We tend to use log2 just to kind of normalize things, and add all of those up. And you get a +1 if you are consistent with the predicted direction of the mechanism, and you get a -1 if your observation is actually inconsistent. So what this helps is that if you have pure noise, hypothetically, you should have a strength score of near 0. But if you have very strong amplitude signaling for a given pathway, like this patient might have, they will have inferred higher levels of this pathway activity compared to, say, patient 2. So just an example of some of what this can do is here, and we're looking at a study in rheumatoid arthritis patients, and these were tissue samples, microarray tissue samples from 62 different patients. We did have infliximab response available to us as measured by EULAR criteria, so let's see. What we're showing you here is on the left-hand side, the strength of TNF signaling in this data set was calculated and shown here. So this is the strength signaling amplitude for this one particular patient. Every bar is a different patient, and it's colored by response or non-response to infliximab. The samples were taken at baseline in the synovium, so it really is the disease tissue. So this individual here has high levels of TNF signaling. This individual here has very low levels of, relatively low levels relative to this person, low levels of TNF signaling. So you can take a population and stratify them for these signaling levels. And what you see is for the TNF strength score that there is an enrichment of non-responders down at the bottom and responders over here at the top. And if you just plot the gene expression changes for TNF, this is what it looks like. And they are kind of all over the map, but the point here is you lose this directional enrichment. So this is an example where there is not this one-to-one correlation between TNF RNA expression and TNF pathway signaling activity. And at Solventa, we did a lot of work on rheumatoid arthritis. And another thing that we did was we actually used these mechanism scores as features to train a classifier to predict non-responders, actually using transcriptomics from blood. And what you're looking at here, kind of the classifier scores for 4 different data sets that were used for training. And this is a cross-validated kind of training test set. And you can see that the non-responders are colored yellow, the responders are gray, and there's an enrichment towards the right-hand side for classifier scores. And actually, this particular classifier was not very good at detecting non-responders, but when it did, it was right, fairly right. So the specificity was very high, sensitivity was low. There's a paper on this if you want to see. And actually, I'll skip this one and just very kind of briefly mention, because I know we have maybe 10 minutes left and I'll leave time for questions, but just to kind of mention what was this work that we were doing at Patients Like Me. So Patients Like Me is still around. It's a pretty wild concept. It's essentially Facebook and social networking for people, for patients. And it provided kind of like a constrained vocabulary and a structure by which patients could report information about how they felt, their symptoms, drugs that they were taking, et cetera, all of this type of information, share that with one another. And there's a lot of really great stuff that came out of that. And in that, Patients Like Me existed for probably like 10 or 12 years before I joined, and I joined in order to bring kind of multi-omics to that equation. So what we ended up doing was building this program called Digital Me, where we were enrolling individuals to, continue to provide patient-generated health data through surveys, but we would also go to their home and we would collect blood from them. And from that blood, we would do some multi-omic profiling. And what that enabled us to have was actually, it was like an observational registry almost. So we would baseline patients, and then every month they would actually take a different survey, and there were different PROs that we would offer to them to take. And then every couple of months, they would get a blood draw, or if there was a change in their medical condition, then they could also like raise their hand and we would go give them another blood draw. So what we ended up having in terms of kind of the end state of this was we had consented like 4,000 individuals. We had baselined about 2,000 of them. We had what I'd say like really good full symptomatic patient-generated health data records for about 1,000 of them. The average longitudinal portion where we had data from them was about a year. And there were like 5,000 total draws completed. So it was a pretty wild time to get that up and running. And we ran DNA, RNA, proteomics, metabolomics, lipids, metals, toxins, something called immunosignature, and also had clinical labs that we hadn't actually run those while I was there, but that was sort of the plan, was to take the blood that we had collected and also be able to get standard clinical labs, which I think would be really useful. And just very, very briefly, we actually spent the 3 years that I was there, we were kind of building up and collecting this data. and unfortunately didn't have a ton of time to be able to do an analysis with it. But one of the scientists at Patients Like Me, Namisha Schneider, she actually did this kind of beautiful interrogation of the lupus cohort that we had. And she had an abstract that was accepted for a talk at ACR, sorry, looking at the ability of proteomics data, specifically Olink, to predict flare response, or flares, to predict flares in the lupus patient cohort that we had. So I think we can jump through that. So I'd say, just in terms of future directions, there's a lot. Like this method is actually fairly established, but not fairly explored, very explored. So there, I think, is really a lot more that could be done with it in the areas that I mentioned before, doing things like drug target mechanism discovery, assessing models for their translatability potential. But something, for example, Sean and I were talking about this morning is, with all of these mechanisms that you're able to make inferences about, can you then start to annotate a patient with what their mechanistic signature looks like, not just their gene expression signature? But I think, in order for all of this to really thrive, is we have to make this type of infrastructure available to the greater and broader scientific community, in particular the biologists, because they're the ones that are doing the interpretation. So that's it. I just would actually like to thank a lot of people, but especially Namisha Schneider and Natalie Catlett, who are like my intellectual sisters in crime and have just been super awesome and inspirational to work with. I've worked with them at Solventa, Patients Like Me, and QuartzBio, so that has just been a delight. And I will leave you with this quote from this fantastic article that came out. I'd say it's a commentary from Ben MacArthur, who is a professor at the University of Southampton, and he wrote this piece called "The Truth and Beauty in Physics and Biology." And the point was that we can learn a lot from each other, but I had to laugh at this quote. So thus, what the physicists may see as an elegant theory, the biologists may see as a facile generalization. And that's it. Any questions?
I kind of have one, Renee.
Yeah.
So you talked very briefly about using the knowledge-based results as the input to a neural network for a classifier. Could you tell us a little bit more about the methodology you think of there and where you think that might head in the near future?
Yeah, it's a good question. It was actually not a neural net. It was, I think it was just like a generalized linear model. And it's a pretty simple classifier. So that was interesting. So we actually did compare it to just using the gene expression data. And the hypothesis we were testing was, if you use reverse causal reasoning almost as like a knowledge-driven feature reduction mechanism, can you get better performance? So, first and foremost, it's nice because it reduces the feature space down, so your risk of overtraining is kind of mitigated. I would say that the problem with this, from a very practical standpoint, is that still in the end, those compressed features that we were using as input represented anywhere from like 20 to like 500 different genes, right? So even at the end of it, I think there were 18 different mechanisms that ended up being, selected by the model. And that represented over 1,000 different genes. So at the end of it, you had a classifier that was made up of 1,000 genes. And that's a lot, like, especially back then when we were doing this, people were not going to use RNA-seq or microarrays as a point of care solution. So we did some work, taking those 1,600 features and really reducing it down to much smaller, more tractable numbers, but it never quite got to like the 6-panel RT-PCR that might have been a more viable commercial solution. But I think, I mean, overall in general, I'm very interested in combining, essentially like trying to figure out how to include prior knowledge as a feature compression unit, or even within an algorithm itself, to help direct maybe the inner workings of some of these deep learning models to the right space, maybe to reduce the compute time required for doing some of those transformations, et cetera. So I think there's a lot that can be done in terms of pulling prior knowledge in.
Hey, Renee. Jim Costello
Hey,
here.
Jim.
How's it
Hi,
going?
how are you?
Good to see you. So
Likewise.
one of the things that some of us on campus think pretty deeply about is obviously you have causal in the causal reasoning, or causal reverse engineering, or whatever you want to call it. That would imply that the experiment design, so the R1881 treatment on the lint caps, the downstream output is fully connected to that one perturbation that you did to the system, right? And certainly there are cell type specific effects, there's interactions with the drug, there's multi-indirect effects, et cetera, et cetera. And I'm very curious on how you think about and try to build in the non-specificity of some of the effects that you're seeing to the actual model itself.
Yeah, that's a great question. And context is everything. So to answer that question from a knowledge representation standpoint, the way that the knowledge is curated is that it is curated actually as an indirect interaction, unless it meets certain criteria to be able to be curated as a direct interaction. So if we are curating androgen receptor signaling, that actually, it's possible that, since it is a transcription factor, that you would curate a direct relationship between the transcriptional activity of androgen receptor signaling and then, a downstream change in RNA expression. But obviously, if you're trying to curate something like TNF, it's always going to be indirect. So, I think way back in 2006 and 7, Metacore, they only looked at transcriptional footprints and not these kind of higher order footprints. So I think that this becomes very important in the interpretation, at the interpretation layer. So, the output of, let's say, a mechanism signature, you might have something like a prediction for PI3 kinase and AKT and PTEN, and NF-kappa B. And that's helpful, right? Because I'm probably going to be more confident about what my transcription factor inference is than my, way upstream controller. So if you just have let's say a growth factor in isolation with none of its downstream effectors that are being inferred or predicted in the model, then I would call that, suspect, right? You can include it in the model, but there should be an asterisk that says, there's a lot of overlap, say, with IL-6, this IL-6 prediction, and the genes that are also predicting TNF. But much more evidence for downstream signaling for TNF than IL-6. So IL-6 is probably really just TNF. So those are the types of interrogations that the biologist does. And those, I would say a big hole in this entire process that could be filled is coming up with better, more semi-automated methods to kind of automatically tease that stuff out. And also our ability to capture knowledge right now is way better than it was 10 years ago. So, when we tried making tissue specific knowledge models and interrogating those, honestly, they just were not large enough to do anything particularly useful, even if we went after it. From the lung stuff. But I think that one of the things that I'm curious about is, I think, is there enough information out there now that we can build better tissue-specific knowledge bases where we can use those exclusively for interrogation.
Cool, thanks. Well, we could talk more about that at our discussion. We got some ideas on that too. So
Cool.
So
Look forward to it.
I guess we're past time.
Yeah, sorry.
That's okay. Tissues. I have one, I think, small question on tissue-specific knowledge bases. So, as just kind of trying to think about context, is your vision in that direction that we know what's expressed in a tissue, and a knowledge base is sort of an intersection of what we know is sort of expressed in a tissue and what is in a knowledge base? Or is it really like this experiment has been done in exactly this cell type and has recapitulated this relationship?
On the second part, do you mean from the data perspective or from what we would curate into the knowledge base?
I was imagining from what you would curate into the knowledge base. Like, is it the curation that changes, or is it some overlay on the curation?
Oh, gotcha. The curation would change.
Okay.
And I wouldn't say that the curation changes. I would say that the abundance of curation, there would just be more, right? So for example, like we wanted to build a very lung specific knowledge base. And so we went and specifically looked at, let's just take canonical like MAP kinase signaling, let's just say, I don't remember if this is the case, but let's say the majority of it was done in, breast cancer cells or something like that. But we really cared about lung. Then we would go and then, look into the literature and say like, okay, well, did somebody in lung cells show that MAP KKK phosphorylates MAP KK? And, usually there was just never enough time to do that for every single project. So like if you found the one that came out of like the breast cancer cell line, like you just take it, especially since you knew it was canonical signaling and it was real, and you wouldn't go look for the lung specific one. But I think with, if we take large scale databases like Expression Atlas, or MSigDB, or the stuff out of, I forget the last name, Mayan lab out of, I think he was a student of Zach Kohane.
Yeah, Bobby,
with the whole
at Mount Sinai.
Yes,
Bobby
yes,
Mayan,
exactly. Yeah, like, their lab has, a whole, a bunch of like molecular perturbation types of signatures where you can actually get, positive and negative directions between the upstream and downstream node. I think like taking big bulk sources like that is going to massively enrich for tissue specificity in a way that like I did not have access to when we were building this knowledge base a decade ago.
Sounds great. I realize we're past time and we've dropped a fair number of people. Is there anyone else who wants to ask a question before we let Renee go? Okay, well, thank you so much for joining us. I know you have more meetings coming up today to chat with folks here in Colorado. And yeah, I'm looking forward to talking. Thanks.